Hi, my name is Hana Trigui from Polytechnique of Montreal, Canada. I am going to talk today about comparison of Legionella pneumophila detection methods in the context of an effective risk monitoring in water system. We will show you our results from a comparative assay and an environmental monitoring in a complex industrial plant. Our experimental approach for lab assay is as follows. We have selected 6 commercially available test methods for L. pneumophila. We have flow cytometer, live depth assay, 2 growth-based method, Legionella from IDEX, which is an enzymatic culture-based method, and filter agar plating. We used also qPCR-based methods, the innovative one online qPCR from BioAlert Solutions, and 2 qPCR kits with DNA extraction. We have GeneDisk and Bio-Rad. We compare the ability of these methods to quantify repeatably defined concentration of a serogroup 1 strain of Legionella pneumophila to mimic the transmissive phase, which is the status of Legionella pneumophila in cooling tower. The strain was grown to post-exponential phase. Then it was responded in Montreal filtered tap water to simulate the makeup of cooling water in Montreal. Prior to quantification of L. pneumophila, it was acclimated for 7 days in tap water of Montreal. 3 defined concentrations were used, 510 to the 4, 510 to the 5, and 510 to the 6. To secure precision of the assays, the experiments were done on 6 replicates over 2 separate days. Here are our results. We have in the left the box plot. It shows in the y-axis the measured concentration of L. pneumophila by the 6 methods, and the x-axis shows the calculated spiked concentration of L. pneumophila. It allows to see accuracy and bias of each method. We have also calculated the coefficient of variation for every method as an indication of their precision. Considering accuracy and precision, Legionella, which is in dark blue, and flow cytometer in red, were the most accurate method to measure Legionella in lab assay. qPCR, which are the 3 methods in the right, slightly underestimate spiked L. pneumophila and are less precise. And as expected, culture was the least accurate and precise. These lab assays are the first in the field comparing these 6 methods together, and it allows us to think about challenges of monitoring in the field. This slide just summarizes what I have just concluded. In the second part of our study, we have monitored L. pneumophila in a complex industrial plant using AFNOR plate culture, Legionella, and Bio-Rad qPCR. Monitoring locations within the circuit included pumps, basin, tanks, and chiller, and it was performed from April to July. Colored boxes means that water sampling was done during the same 40 minutes on the same days, and numbers show different sampling parts. In the red dotted lines show the detection limit of each method. As you see here, Legionella and qPCR show variation between sampling locations and time. When the concentration increased, the method captured this variability as shown here in the box 3 and box 10. We see also that culture-based method and Legionella results are also close to the limit of detection, unlike qPCR, which detected L. pneumophila where culture plate and Legionella did not for all dates. The variability in concentration between sites and time lead us to say that we should monitor a critical process sites with high Legionella pneumophila concentration and aerosol formation. The final slide here shows the results of online qPCR monitoring in the same cooling tower in the 3 pumps of the cooling tower's system. There is a main circulating pump, a secondary functioning pump, which was used when it needed and an offline one. Monitoring was done from May to September. The y-axis shows LP concentration in log units, and the vertical dotted line show the biocide treatment addition. As you see here in the graph, the online LP monitoring shows that levels in the secondary pump in orange are usually higher than the primary in blue. 86% and 37% of the measurement of in the secondary and offline pumps, the orange and green, exceeded 10 to the 5 genomic unit per liter. The secondary and offline pumps are unresponsive to the treatments. Knowing that the monitoring plan in this system, cooling tower system, is to measure Legionella by plate culture monthly. So this plan cannot capture the variability of LP concentration in the different circuits of the cooling tower. And as you see here, the online monitoring is the best suited for quick response process since we have the results during the day, and it can detect high risk situation in the cooling tower system in near real time. Finally, I have to thank all my team in Michel Prévost Lab, and thank you for your attention.